This is the first cell of a multicellular organism. It contains all the information needed to build this organism. But, if the cells simply divide to create copies of themselves, we will end up with just a clump of identical cells. However, we want these cells to form different tissues and organs in the body. More than that, we want it to be not just a lump of different tissues, but also have the right shape. To understand how this happens, I began a new project simulating how the cells of a multicellular organism interact with each other. But first, a bit of theoretical background. It's a DNA molecule. Sometimes a gene is found on it. A gene contains instructions for producing a protein or functional RNA. Depending on which set of genes are active in the cell, we get different types of cells. In addition to genes, there are also various regulatory sequences in DNA that regulate the work of genes. These regulatory sequences themselves are controlled by the active molecules that are present in the cell. In this project, I'll refer to them as morphogens. Usually, they are proteins or RNAs. They can either be produced within the cell itself or enter from outside. As a result, we can get complex gene regulatory network where different genes influence each other. By using positive and negative feedback and time delays, it's possible to create triggers, filters, oscillators, timers, and others. Several morphogens may be required to regulate a single gene. The same morphogen can act both as an activator and a repressor depending on the conditions. A gene can not only be turned on or off but also have its activity regulated. At the molecular level, regulation looks quite intricate. The DNA molecule can bend, so regulatory sequences can be located far away from the gene they control. This makes it difficult to study them. Moreover, regulation can occur not only at the stage of reading information from DNA but also at later stages. There are several ways for cells to communicate with each other. Today, we'll use the option where a cell releases active molecules into the intercellular environment, creating a concentration gradient around itself. In future videos, we'll use the first option as well. The range isn't very large. In my project, I've limited it to 9 cells. Neighboring cells can react not only to the presence of a morphogen but also to its concentration. The exceedingly complex gene circuit that specifies a portion of the developing sea urchin embryo. Each colored small box represents a different gene. Those in yellow code for transcription regulators and those in green and blue code for proteins that give cells of the mesoderm and endoderm, respectively, their specialized characteristics. In the video, for the sake of importance, I'm using images from this book. In my project, everything has been greatly simplified and reduced to a simple scheme. Additionally, my cells have a fixed size and position on a hexagonal grid, so cell division can only happen along the edge of the organism. That means my simulation is quite distant from what actually occurs in real organisms. My main focus was on the interaction between cells, so sometimes I see this as a swarm intelligence project. Today, let's try to create something similar to Placozoa, with the ability to regenerate and reproduce. Here is a list of the genes we need for the project. The first 14 genes produce the morphogens necessary for regulation. They are all numbered according to the gene number. By default, they are inactive. For them to work, certain morphogens must be present in the cell. It's worth noting that morphogens degrade over time, so they need to be constantly produced to maintain the necessary concentration. For convenience, I've divided them into 2 groups. The first 6 genes will produce a morphogen that doesn't leave the cell and activates the same gene. Let's record this in the regulatory sequences. This creates a positive feedback loop, where the activated gene sustains its own work. I can use these genes as memory, and I'll sometimes refer to them as triggers. The following genes will be necessary for interaction with other cells. Each of these genes produces its own morphogen, which can penetrate into adjacent cells. When activated, you need to specify how strongly the gene expresses, meaning how far the product of its activity will spread across neighboring cells. The maximum distance is 9 cells. The next 2 genes start timers. When a gene is activated, you need to specify the duration of the timer. At the end of its run, the corresponding morphogen will appear in the cell. If the cell divides, the timer remains active only in the mother cell. All cells divide when there is such an opportunity. By keeping the next gene active, the process of cell division can be blocked. Activating the next gene leads to apoptosis, meaning the cell kills itself. When activated, you need to specify a number inversely proportional to the probability of this happening during one step of the simulation. The following 2 genes trigger a cascade of genes that produce cellular differentiation. Let's look at this in a little more detail. Today we will use 3 types of cells. Germline cells or stem cells. In reality, they differ, but in this project they are the same cell. And then there are 2 types of somatic cells. Flesh cells, which perform the primary task of providing resources for the organism. Skin cells whose main function is to protect the organism from the environment. Each of these cells, when dividing, produces a cell of the same type. However, a stem cell can transform into any other type of cell. Complex transformations take place, unnecessary genes are blocked through methylation, and returning to the state of a stem cell or transitioning to another type is usually no longer possible. In reality, differentiation can occur in multiple stages, as shown in the image using blood cells as an example. Each cell type has its specific morphogen, which we'll use to identify the cell type during regulation. Activation of the last gene causes the cell, if possible, to move away from the 12th morphogen. Here are all the morphogens that we will need. Let's add another morphogen, which indicates the number of neighbors a cell has. To describe the regulatory sequence, I'll use this method. If it's a stem cell, and the 6th and 10th morphogens are present, and the cell is fully surrounded, activate the gene. If the gene needs to be activated with a specific intensity, we'll indicate that after the condition. If you need to invert the conditions, then it is written on a pink background. That is, the gene will be activated only in the absence of the 6th morphogen. Now it's time to create our first organism. If we initiate cell division now, we will get a growing mass of identical cells. It is necessary to limit the growth somehow. Let the first cell secrete a morphogen, which will serve as a growth boundary. The first naive solution. Let the cell, if it is a stem cell and has no neighbors, begin to produce a 6th morphogen for a range of 7 cells. But as soon as the cell divides, the conditions will no longer be met, and the production of the morphogen will cease. Let's go the other way. The absence of neighbors will turn on the zeroth gene, which will be used as a trigger, and the cell with the active trigger will produce the 6th morphogen. This won't work either. The zeroth gene will be active in every cell after division. In this project, asymmetric division only works for the timer, and the timer will only function in the parent cell. Therefore, we will first start the timer. And upon completion of its work, the timer will activate the zeroth gene. If the timer duration is set too large, the dividing cells will leave the morphogen's area of influence. If it's too small, the first cell won't have time to be surrounded by neighbors and will also start dividing. We'll keep the timer duration short and introduce a ban on cell division for those that have the zeroth gene activated. Now everything is working correctly. All that's left is to add a restriction on cell division for those that have moved beyond the range of the 6th morphogen. And nothing starts. The first cell refuses to divide because there is no 6th morphogen. We'll have to adjust the conditions slightly. For cell division to stop, not only must the 6th morphogen be absent, but the first gene must also be active. And the first gene will be activated by the sixth morphogen. Now there's nothing stopping the first cell from dividing. At the end of the timer, the sixth morphogen will begin to be produced, which will then activate the first gene in all cells. Upon reaching the boundary of the morphogen, the cells will stop dividing. It's time for cell differentiation. Once they reach the boundary of the sixth morphogen, the cells will start turning into skin cells. We'll prohibit skin cells from dividing. Before transforming into a skin cell, each cell will release a small amount of the seventh morphogen. This will serve as a differentiation signal for the other cells. Sometimes I change the color scheme of the morphogens to better visualize their operation. The seventh morphogen will trigger the transformation process into flesh cells for those under the influence of the sixth morphogen. Only the central cell with the activated zeroth gene will remain in its original state. It will become the germline cell for producing future organisms. We'll restrict the division of flesh cells in the absence of the sixth morphogen. The skin turned out to be perforated. Currently, the organism is capable of generation. If the central cell is removed, all stem cells will turn into skin cells due to the lack of the sixth morphogen. To complete cell differentiation, we'll add a wave of the seventh morphogen. Stem cells, upon receiving the seventh morphogen, also release a portion of the seventh morphogen and transform into flesh cells. A wave of transformation sweeps through the organism. Here, for some reason, 2 germline cells remained, but that's perfectly fine for us. The skin is perforated and does not heal when damaged, so we'll need to fix that somehow. Let's add an additional condition to the differentiation process into a flesh cell. The cell must be completely surrounded by other cells. This allows the skin to form without holes, but it doesn't solve the regeneration problem. We'll come back to this later. Now we're going to make significant changes to the genome to make the organism larger. The first cells that reach the boundary of the sixth morphogen will become germline cells. To do this, we'll activate the zeroth gene in them. In response, they'll emit a burst of the eighth morphogen. The eighth morphogen activates the second gene in all cells except the germline cells. To transform into skin now requires the activation of the second gene. Those cells that were the first to reach the boundary of the sixth morphogen become germline cells. Using the eighth morphogen, they disable this privilege in the rest of the cells by activating their second gene. Cells with the active second gene, upon reaching the boundary of the sixth morphogen, transform into skin cells. We've got a perfect organism. The skin protects it from external influences. Flesh cells gather resources and energy and send them along the gradient of the morphogen to the germline cells. Flesh cells are able to regenerate, but if a germline cell is removed, some worker cells find themselves without a purpose and don't know where to transfer the collected resources and energy. We'll monitor the figure and get rid of such cells. Now, in addition to the sixth morphogen, germline cells will also secrete the 13th morphogen, but at a slightly lower intensity. Flesh cells will stop dividing when there is no 13th morphogen, and will die in the absence of the 6th morphogen. Flesh cells no longer intrude where the skin should be. If a germline cell is removed, all associated flesh cells die. The skin still needs some work. Let flesh cells that have fewer than 6 neighbors start secreting the 9th morphogen. Skin cells, under the influence of the 9th morphogen, gain the ability to divide. Now the skin has the ability to regenerate. Beautiful scars in the form of thickened skin remain. But if the central cell is removed, a huge, unnecessary growth of skin cells forms. Each flesh cell will secrete the 10th morphogen over a distance of 1 cell, and a skin cell dies in the absence of this morphogen. Regeneration works well. After regeneration, the skin becomes smooth and silky. And after the death of the germline cell, everything is neatly sealed up. It's time to think about reproduction. We have several germline cells, and all other somatic cells must die. We will use the 11th morphogen for this. Suppose one of the germline cells has accumulated enough resources to create a new organism. Its third gene gets activated. I'll do this manually. The third gene will launch a wave of morphogen and deactivate itself. On the right side, I'll record the regulatory sequences that deactivate the gene. To ensure the morphogen wave travels throughout the entire organism, every cell reached by the 11th morphogen will also release it. When the organism has amassed enough resources to create new copies of itself, I activate the third gene in one of the germline cells. A wave of the 11th morphogen swept through, triggering apoptosis in all somatic cells. The remaining cells have the 0th gene activated, so they cannot divide or produce the 6th morphogen. Additionally, the 11th gene is operational, which activates itself. Here, we've created positive feedback. Let's remove it. Now, only somatic cells will produce this morphogen. Also, let the wave of the 11th morphogen deactivate all functioning triggers in the germline cells. Launch the apoptosis wave. Note that the germline cell divided without starting the timer or activating the necessary triggers. The same thing happened with another cell. That means the development process of the new organism was disrupted from the very beginning. As a result, we ended up with just a bunch of dividing cells. To kickstart the proper development of this organism, the first cell shouldn't have any neighbors. So, for now, we need to temporarily block the division of germline cells. Activation of the third gene will also activate the fourth gene. A cell with the active fourth gene will secrete 12th morphogen. The 11th morphogen, which causes apoptosis in somatic cells, will also activate the 5th gene. It will block cell division and the initiation of the 0th timer. In the end, we are left with germline cells. The active 5th gene blocks their further actions, while the cell that initiated the replication process releases a morphogen that sets the direction for all cells to disperse. Final touches. In the germline cells, the first timer is also activated for a long period. At the end of the timer's run, it deactivates the 4th and 5th genes. Deactivation of the 5th gene unblocks the 0th timer, which initiates the formation of a new organism. While the 5th gene was active, all single cells would move away from the 12th morphogen. We've successfully created several new organisms. Theoretically, this is already a solid foundation for creating a simulation of artificial life, but I have different plans for now. All the cells in a multicellular organism are clones of a single cell. The vast majority of them are initially destined to die, and their main task is to provide resources for the germline cells, which will pass the genome on to subsequent generations. Remember that all cells contain the same genome. Having only the genome, it is nearly impossible to understand what kind of organism it is. You can only find out by launching the development of the organism. I want to draw your attention to the fact that the interaction of cells within a multicellular organism is fully analogous to swarm intelligence, where multiple agents, sharing the same algorithm, achieve something greater through collective activity than what could be seen in the algorithm alone. In the next video, let's try to create more complex organisms that represent not just a shapeless spot, but have a sophisticated form. To do this, we'll need to give the cells new capabilities. We'll experiment with random mutations in the regulatory sequences and see where that leads. With the same set of genes but activating them differently, you can obtain numerous distinct organisms. This raises the question, what's most important in the genome? Be sure to like, subscribe, and leave a comment. A special thank you to those supporting me on Patreon. Bye for now.